Alright, pre-AP, we just got done with transcription. Now we're headed on to translation. So, remember in transcription, we took DNA as a template and we made it into mRNA, messenger RNA. Now, after we've gone through transcription and we got all the way through the steps, your RNA polymerase read your DNA segment, and it made mRNA by attaching nucleotides such as A, U, G, and C. Now we're ready to move on. That mRNA got modified at the very end. We took a 5' cap and a poly A tail and put it on either side. So this would be my 5' cap at this end. At the very end, we have a poly A tail. Remember, these are to help protect the mRNAs. They're going to leave the nucleus. They're going to leave their home in the nucleus, and they're going to go out into the crazy world of the cytoplasm. And if they didn't have that 5' cap and that poly A tail, probably some kind of digestive enzyme out in here. It's a cruel world. Probably disintegrate it and, hurt it, destroy it. Then we'd have no proteins. So you're probably going to ask me, well, where do they leave the nucleus at, Miss Miner? Remember, your nucleus has pores, has these little openings. And the reason for that is so that mRNA can leave and be able to go to the cytoplasm, meet up with a ribosome, and complete the last step of protein synthesis, translation. So remember, hope you still have that highlighter and that pen. We're going to need it. So we're to part 3, translation. We have copied the recipe. We have read the recipe. If you're going to make pancakes after you've copied the recipe, you've read the recipe, you've gotten all your ingredients together, that flour, that butter, that sugar to make pancakes, you are now ready to make the recipe. And what we're going to do now is we've went from DNA to mRNA. We're going to take that messenger RNA that we just made and we're going to turn it into an amino acid chain. Remember, that is eventually going to fold up into its proper shape and become a protein. So please put that out there beside translation. Alright, so highlight. Translation, the process of actually making the protein. Remember, we're going in order. Big letter A, you need to know this occurs at the ribosome. Ribosome's kind of like your translator. He's going to translate this message into something the cell can use, an amino acid. So again, we're going to take our mRNA into a primary sequence of amino acids for making protein. Remember, protein aren't protein until they become that squiggly shape. This is not a protein. That's just a string of amino acids. This is primary structure, remember? This is going to be tertiary or quaternary structure. Alright, so let's highlight. We're going to need the assistance of a tRNA. That's why I made you know mRNA. tRNA stands for transfer RNA, because this job is to transfer free amino acids from the cytoplasm to the construction site of the ribosome. So again, we're going to need that amino acid chart again, guys. It does not go away. It stays here the whole unit. Transfer RNA, just a close-up. It's actually in the shape of a T, too. How cool is that? So we need to highlight that little letter 1, where it says, remember, the anticodon. Remember, that's the opposite of your codon. He's going to be right down here. And he's going to read this mRNA strand. And he's going to match up his anticodon with the codon found down here. Remember, codons are 3-letter sequences. And at the other end, this is where an amino acid is going to attach. It would probably be a pearl shape. It's going to attach up here at the very top. Down here at the bottom is the anticodon. This one's A, G. And it would match up with a codon down here that was U, C. He would know, whoa, my anticodon matches up with you. I'm going to go get my amino acid up here that codes with this anticodon, and I'm going to attach it and start forming a chain. Another tRNA would come in, read this guy. He'd have an amino acid, and he'd attach it up here eventually. He'd have an anticodon that matched this codon, and then we would start building a chain. Just as a kind of, ooh, this is what's going to happen in a minute. So highlight the anticodon matches the codon of the mRNA molecule, ensuring the proper amino acid is brought to the construction site at the ribosome. Remember, if they don't match, it's going to be the wrong amino acid. Can't happen like that. So let's go down to ribosome structure. Ribosome structure. We just need to know that this is made of a large subunit and a small subunit. So let's just draw us a ribosome. It's going to be kind of this big blob. And on the bottom it's going to have a smaller blob. So go ahead. This is large. Subunit. This is small subunit. That A, P, and E site, that's just to show you that, hey, ribosomes have very complicated places. I'm not going to ask you that. If you want to know, the P site is the binding site for the polypeptyl tRNA. E is the exit site, but I'm not going to ask you to know that. And then, the only thing that I need you to highlight is under ribosome structure, it has 2 parts. One, there's a small subunit. It's the platform for the work. It's kind of like your desk at school. Number 2, it has a large subunit. This is the factory for making protein. This is where your mRNA would fit. In between the two, like a sandwich. And then 3, the ribosome translocates. That's a fancy word for walks down the mRNA strand, 1 codon at a time, 3 nucleotides at a time, 3 letters at a time, until it gets to a stop codon. It's going to come in, it's going to say, hey, tRNA, I read my codon. Start building me an amino acid, yo. Once he deposits his amino acid, he can go to leave. Kind of like if there's a bunch of people that wants to go to the bathroom. I have a better picture of this. Ooh, here it is. This tRNA comes and he comes and sits down. Let's start with this one. This guy came and sat down. The one that's leaving right now. We're going to explain why he's leaving. He came and sat down. He matched up his anticodon with the codon on the mRNA strand. He lined it up as the ribosome read this mRNA strand. He said, hey, tRNA, I need an anticodon of a CCC. And he said, okay, boss. So he came and he sat down here. He had some amino acid attached at the top, and he deposited it. Well, he said, okay, tRNA, you deposited your amino acid, you're done for the day, or for right now, I'll call you when I need you. And he says, okay, boss. And then he's going to leave, and go back out into the cytoplasm until he's needed again. But, whoa, there's a step we missed here. He can't leave yet until another tRNA comes and sits down. It's kind of like if you need to go to the bathroom and there's a long line and you ask me, can I go to the bathroom, Miss Minor? And I'll say, well, so-and-so's already went to the bathroom, but whenever they come back, I'll let you go. This tRNA sat here for a while. He did. He deposited his amino acid. The ribosome said, great job, you can go. But you have to wait till another tRNA comes and sits down. Then you can go. You have to take turns of your tRNA. This tRNA right here can't leave until this guy comes and sits down. He deposits his amino acid, and then he may leave. This guy wouldn't be able to leave until another tRNA came and sat down, read his codon, deposited an amino acid, then he could leave. We take turns in the cellular world, just like in the ninth grade world. Alright, so last thing, number 4 on there, remember, your ribosomes are not membrane-bound organelles. They are just cell structures. Alright, let's talk about the process. Remember, it's going to be initiation, elongation, termination, just like transcription. So initiation, just know this is the building, the factory, needed to make the protein. So, what happens is, this small subunit right down here, it's going to attach to that 5' cap. Then, this sends out a signal to this large subunit. It says, hey, I've attached to this 5' cap down here. We're ready to make a protein. Then, what it does is AUG, the start codon, brings in a tRNA. And then, this is attached to, because remember, AUG is the start codon, and it codes for the amino acid methionine. And this starts the production of a protein. Let's highlight the large subunit is aligned with the methionine up here in the P site. That's why we can't see it. It's covered up. It's sitting in the P site. And then the A side is open over here for the addition of the next tRNA. Oh yeah, because remember guys, he can't leave until another tRNA comes and sits down. This is open. It says, hey tRNA, I've read another codon. Come sit on down. Then he's going to deposit his amino acid, whatever that one is. Then this guy can leave. Taking turns. So that's initiation. We are starting the process. Elongation. Please highlight that this is the actual making of the primary sequence of an amino acid. All that means is the ribosome walks down the mRNA one codon at a time, saying, hey tRNA, here we go. We've already initiated. It's saying, hey, I've recognized another codon. You come and sit down. You can leave. Figure out another polypeptide. He comes and sits down. Attaches. You can leave. Find another one. Remember, he sat down, so he finally gets to exit the E site. He just scoots on over. So, and then the last thing, elongation, we're just keep on building that polypeptide. Termination, remember terminate means to stop. So termination occurs in the termination codon, the stop codon, whenever it reaches the A site. So remember that A site over here, where that little yellow piece is. We call that a releasing factor. It says, okay, we've read everything. We may stop now. So let's highlight that. A releasing factor. It's an enzyme. It enters that A site, causing the hydrolysis. Hydrolysis, breaking water. Reaction to occur that releases the protein from the last tRNA molecule. After hydrolysis occurs, the protein detaches from all the subunits. That's what's happening over here. It's detaching from everything. Large subunit's going to go his way. Small subunit's going to go his way. This new polypeptide right here, it's released. It detaches. And then, number 4, let's highlight. mRNA may be reused to make more particular proteins, or it could just be broken down. If you need more of that particular protein, you're going to keep reading it on a different ribosome. And then post. So let's go to Roman numeral 2. Post translation modification. We got to modify some things. This is going to be a big deal. So let's highlight this. Please know, I forgot this. Go to the parenthesis after translation modification. All this means is the protein folding must occur. You got to fold this primary structure if you want the protein to be functional. Remember, proteins don't function if they look like this. They only function if they look like this. Remember, proper shape, proper function. This is primary structure that we just made. We want to take this and turn it into a tertiary structure or quaternary structure. So this is how we do that. Let's highlight big letter A. The primary sequence enters a chaperonin, which is just a folding protein. And then what it'll do is the protein, it will stay inside the cell. We go to big letter B. The primary sequence again, it'll enter the rough endoplasmic reticulum to fold. So the chaperonin is kind of like a chaperone. It kind of says, come on, little primary structure, I'll protect you from this harsh, cruel world. And I'll take you over to the endoplasmic reticulum, and you're going to fold into the protein you need to be. And then we'll export you out of the cell. So chaperonin, there's a picture. Your polypeptide goes in. He gets all squishied, folded up correctly. Then he pops out of the chaperonin. He's correctly folded. We're going to send you to the rough ER. You're going to get further shaped. Then we're going to ship you out through the Golgi apparatus. So there it is. Goes into the ER. Then it will leave. So part 4, this is your mutations. What if something happens? Codon chart yet again. You get a wrong amino acid, you get the wrong protein. Wrong protein, wrong function. So point mutations. Let's go ahead and highlight what a mutation is. Let's highlight. Mutations are changes in the nucleotide sequence of DNA or mRNA. Remember, DNA makes mRNA. mRNA codes for proteins. We call these, they're caused by mutagens, which is either a physical or a chemical interaction that changes the nucleotide sequence. And there are many examples. I just gave you UV light, cigarette smoke, alcohol in excess, viruses, car exhaust, chemicals. Maybe you worked in a nuclear power plant. Maybe you work in high amounts of radiation at your job. These can change your DNA sequences, which could potentially change your protein sequences. So there are 3 major types. Please know that a point mutation, highlight this. This is just a single nucleotide that mutates, and it affects a single codon. Remember, codons code for amino acids. A large, folded up string of amino acids is a protein. So a silent point mutation, just know that it causes no change in the amino acid. We'll never know because, well, it doesn't affect you. So they're okay. Missense point mutations, this is a mutation that changes the amino acid it codes for. We call it a mistake. This is what causes sickle cell. Please highlight that. And a nonsense point mutation, this changes from coding for an amino acid to coding for a stop protein, a stop codon, which means there will be no protein made, which is, I like to call it's nonsense. Doesn't code for a protein. Doesn't code for a protein, you're in trouble. And then our last one, reading frame. Just know that this is your DNA, will highlight your whole DNA sentence or sequence is changed behind the mutation. So this is whether, so you can write what it says at the very top, nucleotides are added or deleted in the sequence. You could have a change in one, like a point mutation, or you could just have something inserted, deleted. It's still bad. So insertion, know that this is adding a nucleotide. And then I gave you some sentences. The big tan dog ran. And then, oh boy, we kind of put in a letter there that wasn't supposed to be there. And then the whole sentence doesn't read correctly. I can't even read that sentence. That's bad. And then deletion, we take out a nucleotide. We deleted one. For example, the big tan red dog ran again. But we took out the I and big. And again, can't even read that sentence. So then go ahead, read big letter D about gametes and somatic cells. We're going to talk about this a little bit more in the next unit, but go ahead and read it. And I will see y'all tomorrow.